Hi everyone, I'm Dr. Safa Khalid, Associate Professor of Hematology and BMT, Asyut University, Egypt. Today I will talk you about video abstract of manuscript entitled Anticipation of Relapse and Acute Graft-versus-Host Disease after Allogeneic Peripheral Blood Stem Cell Transplantation. The fundamental rule of the antigen-presenting dendritic cells. This article is published at Journal of Inflammation Research. This is the research group of this study. Background and aims. Dendritic cells are antigen-presenting cells. Efforts to explore the role of disease in acute graft-versus-host disease after allogeneic peripheral blood transplantation are gaining traction. However, further research is needed to identify the particular lineages and their values in terms of developing an evidence-based acute graft-versus-host or relapse preventive strategy. In this study, we monitor DC counts and subsets in the peripheral blood stem cell graft during harvesting stem cells in recipients to elucidate their value in anticipating disease relapse or acute graft-versus-host disease. Now, methods. We enrolled 29 participants using FACS technology. The total count per kg of CD34 plus DC cells and DC subsets were analyzed in the 29 graft components using CMR444, CD11C, and CD4 monoclonal antibodies. Results. In the 29 grafts, we detected a significant positive correlation between DC1 and DC2. Significantly higher counts of DC and DC1 in those who develop acute graft-versus-host disease were also observed. Relapsed cases were also associated with higher counts of DC and DC2. A significant positive correlation was recorded between DC and DC1 counts and the day of myeloid engraftment, as in Figure 1. Conclusively, this study showed higher donor DC counts are linked to the incidence of acute graft-versus-host disease and DC2 with relapse. Now this figure shows results of the study, and thank you for listening.